Hey, how's it going, class? So today we're going to teach you everything you need to know about the genetic code and DNA until I teach you the next one about more stuff you need to know about DNA. But so far, that's what we need. Now, the thing we need to understand is that DNA, we've probably seen this famous image before. This is actually a really terrible one. Sorry for that. But you've seen this before, the double helix of DNA. It's just that spiral staircase that it makes. And in each one of these, you see these little rungs, these little steps on it. Well, these steps, as you remember, are made up of two different nitrogen bases. Now, when I say there's two different nitrogen bases, there's four that I can choose from. These are four different chemicals. The first one we're going to run into is adenine. The next one is thymine. Also, we run into guanine. And lastly, we have cytosine. Now, with these four bases, they have a very particular way of how they pair with each other. Remember I said about that imaginary friend we keep talking about? Because, we're smelly and have no friends, so we can only be friends with our imaginary friend named ATGC. Remember, it's ATGC. So that means that wherever you have an A on one side, you're always going to find a T on the other side. Or if you have a T on this side, you're always going to find an A on this side. But if you have a G on this side, you're not going to find a C connected to it. Instead, you're going to find a G connected to it. And that's how DNA is read. So let's straighten this out and take a look at it. So let's say we have our DNA right here, and we have it like this. And we remember that this is one part, and this is a different part. This is one chemical, this is a different chemical. So if this side is made of adenine, this side would have to be made of thymine. If this side was guanine, this would have to be cytosine. And even if it was reversed, if this side was cytosine, this side would have to be, you said it correctly, guanine. And it goes all the way through like that on DNA. So how scientists read that is they look at that a different way than having to draw this whole thing out. That would take too much. So instead, when they're writing out DNA, they'll simply do it like this. So as they look at the DNA, they'll make a list. So they'll read the rungs, and let's say it says A, T, T, C, G, A, T, C, C. And they see it like this because on one side of the DNA they see a rung that's made of this. And this, remember, this is only half of it. So it's only if you had one strand it would look like this. Again, you notice that's different than if you had the ladder, which is like this. It only has one half side. Now, on the other side, we only have to look at this one side to know what the other side looks like. Because again, if we remember, A always goes with T, T always goes with A, G, C, T, A, G, G. And we see that that's how they're going to match up. So if I ever give you half a strand of DNA, you'll know what the other half of the strand of DNA is. And so it's actually something quite simple. And this is actually what they can do when they look at people, because all of our DNA is very unique. And the way you can prove that the DNA is unique is look at you and your classmate, the people you sit next to. You're both human, you both have 2 eyes, 2 arms and all those things, but there are still some differences between you. Your eye color, your hair color, how tall you are, how short you are. And your DNA is the thing that codes for all this stuff, that tells your body how to look the way you do. And if you notice, no two people look exactly the same. They can look very, very similar, very close. And you get celebrity lookalikes and stuff like that, but never 100%. Actually, the only people that will share 100% the same DNA, as long as there's no mutations in there, and we'll talk about that later, are identical twins. Now again, I don't think we have any identical twins at our school. You can correct me if I'm wrong, but I'm pretty sure in middle school we don't. I don't think anybody's identical. Everyone's fraternal, so they do have changes in DNA. It's not always exactly the same. But if you have an identical twin, it is 100% the same, again, unless there's a mutation. Now, the other thing that we're going to cover real quick is just what happens when DNA decides that it needs to double itself. Now again, remember we talked a lot about DNA replication. And that's the process where you have a DNA strand like this, and remember it's making that double helix, but instead we decided to straighten it out to make it easier for us. We have a strand of DNA that we took off the double helix and just laid it out straight. And it's getting ready to go to mitosis, or the first part of meiosis, but we'll do mitosis for this one. And remember, the first thing it does is it has to copy this DNA, because as it makes two cells, if we only had one, then one cell would get DNA, the other one wouldn't, and then you'd have a problem because the other cell would have no blueprints on how to operate and would eventually die. So what it does is it goes through and it, oh, that's a terrible tear. It starts to tear or unzip. And you'll start to see something like this. So some of the ends will still be connected, but slowly they'll all start to come apart. You can see that there's gaps in here. Now the thing is, inside your nucleus, where you still have, where your DNA is still sitting, is you have these free-floating little chemicals. Adenine, and you have thymine, and you have guanine, and you have cytosine. And you have things that will come take these and put these in the correct places. So let's say this one has to go here, this one has to go here, this one has to go here. And what you're going to see actually is that as it unravels, it'll find a new spot to put these. And it'll make its own backbone. And then you'll find that what it'll do is it'll actually start making two different strands. Now it does this very fast. Your body's really efficient at doing this. So as it unfurls, it starts putting these new strands, and you'll see this thing grow out further and further. And eventually you'll end up with two completely different strands. Well, they're not different, they're identical, but they're separate strands of DNA. This one will go to one of the cells, this one will go down. So let's say this one goes to the old cell, this one goes to the new cell. Pretty cool, huh? That's DNA replication. And if you need a good visual of how it's working and all the parts popping into place, what you can do is you just simply look on page 112 and 113. And again, remember, if you have any questions, just ask me. When you're looking at that, look at it's in the top right corner on page 113. You can actually see a magnified DNA strand. Now, again, you can see that we can't really get close enough to see really good detail, but you can actually see the DNA opening up and starting to form two completely separate strands, so it's pretty cool. So, all right, that's all I have for you right now. Until next time, take care, and hope studying goes well.